I'm Dr. Sumati Hajela, Associate Professor, Department of Biotech, IPS Academy, Indore. Topic of my lecture is Medical Biotechnology is Advancing Modern Healthcare. Then what is medical biotechnology? Medical biotechnology is the use of living cells and materials for research and to produce pharmaceutical and diagnostic products that help treat and prevent human diseases. Some of the latest areas of medical biotechnological advancement include pioneering work in genetic testing, advanced drug treatments, and artificial tissue growth. There are many applications of biotechnology, but these are the main applications which are helpful in medical field. So biotechnology is helping in the field of stem cell treatments, monoclonal antibodies, genome sequencing, gene therapy, and CRISPR technology. As for stem cells are undifferentiated cells of animals and plants. They are found both in animals and plants. They are the continuous source of the differentiated cells that make up the tissues and organs. They have two important characteristic features. First, they are unspecialized and capable of renewing themselves through cell divisions, sometimes even after long periods of inactivity. When a stem cell divides, each new cell has potential either to remain a stem cell or become another type of cell with a more specialized functions like muscle cell, red cell, brain cell, lung cell. All the cell varieties present in the body are produced by stem cells. Their second characteristic feature is that under certain physiological or experimental conditions, they can be induced to become tissue or organ specific cells with special functions. So main function is that they serve as a sort of internal repair system, dividing essentially without limit to replenish other cells as long as the person or animal is still alive. For example, our intestinal cells daily shed, and there is a complete set of those cells are present on the daily basis, which are produced by stem cells present in the intestine. Similarly, we can see that our skin sheds and new cells take their place. So this function is performed by stem cells present in skin. Then what is the potential? Given their unique regenerative abilities, stem cells offer new potential for cell-based therapies. Cell-based therapies are also known as regenerative or reparative medicine. It helps to treat diseases like diabetes and heart diseases, etc. Existence of hematopoietic stem cells was demonstrated in 1975, and till 2016, we knew that there are few varieties of stem cells exist. First variety is stem cells, which are totipotent. Totipotent means these are present in the morula state of zygote, and these cells are capable of making placenta and all the complete individual. Embryonic or pluripotent stem cells, these are present in blastocyst, and it can make uh, all the cell types in our body. All three germ layers are produced by these cells, but these cannot make placental cells. Then adult cells, adult stem cells or somatic stem cells, these are produced from these cells, pluripotent cells. Still, they have characteristic features of stem cells, and they can make cells of various organs. Wherever they are present, they will make that tissue. And finally, induced pluripotent stem cells. Now, induced pluripotent stem cells are the variety of cells which are induced to make embryonic cells first, then under the influence of several factors, they can make different variety of cells again. So these are induced. These grow into adult cells. These are under certain conditions, they are reverted back to embryonic stem cells, and from here they are directed to make different variety of stem cells or different variety of adult or somatic cells. Here we in this diagram shows the same thing. This is the oocyte and or zygote, and this divides into modular stage, and the cells are totipotent. They can make placenta, and uh, all the three germ layers which occur in the embryo. Then this is a blastocyst. It, it carries this inner mass. This mass is pluripotent stem cells which can make circulatory system, nervous system, immune system, and nervous system. Nervous system, it is shown for nervous system only that now it contains unipotent. Similarly, other systems will also contain finally unipotent cells which can make only that cell where they are present. And cell division, you can see number one is symmetric stem cell division, and number two shows the asymmetric stem cell division. Number three is the progenitor cell. Progenitor cell now is committed to make one particular type of cell, so it divides and gives rise to adult somatic cell. And number four can be said as terminal differentiation into mature cells. This figure is taken from Wikipedia. Now, in 2017, the world famous Salk Institute, one of the leading non-profit medical biotechnological organization, created a new kind of stem cell which is known as extended pluripotent stem cell. This EPS helped to grow mice, complete mice, in a laboratory from an embryo. So it had the capability to make placental cells also, which we can see here that EPS cells can make muscle, blood, bone, tissue, other tissues, among many others, and most importantly, placenta. Placenta is extra embryonic tissue needed for embryo to survive and grow. And I told earlier that even embryonic stem cells are pluripotent and cannot make placenta. So this was an achievement that scientists could make EPS cells which can make placenta, which the cells which are capable of making the complete organism. Stem cells can be used for disease modeling. Here, uh, according to this scheme, diseases can be modeled. Otherwise, we use animals as disease models, but in vitro also, and now it is possible to model a disease. For that, mouse embryonic stem cells or human embryonic cells are taken from the inner mass from blastocyst, and induced stem cells can be taken from their source will be biopsy tissues which are taken from uh, diseased uh, patients, and under the influence of reprogramming factors, all these three varieties of cells are capable of making somatic cells of all three germ layers, and it can be grown into muscle cells of various varieties like smooth cell, smooth the muscles or cardiac cells and neurons. All the variety of cells can be grown, and while IPS taken from biopsy tissue will act will serve for diseased tissue, and these mouse and human sources will serve as control on normal tissues, and then various experiments can be uh, uh, made, can be started to find out the cause of disease or drug action on disease and controls conditions. Various type of experiments can be done with the help of stem cells in in vitro conditions without slaughtering or sacrificing animals. Diseases and conditions where stem cell treatment is being investigated. We can see it is tried for a stroke, traumatic brain injury, learning defects, Alzheimer, Parkinson's disease, missing teeth, heel wounding, bone marrow transplantation, spinal cord injury or osteoarthritis, rheumatoid arthritis, Crohn's disease, multiple site cancers, diabetes, muscular dystrophy, myocardial infarction, amyotrophic lateral sclerosis, deafness, blindness, baldness, etc. So for everything, stem cells are tried and uh, used for make drug therapy. Uh, for COVID treatment also, we found that, or rather researchers at China found that it is useful, though um, in the current scenario, much test was not carried out still. Uh, they reported that 7 patients were treated with stem cells at Beijing Yuan Hospital. Each patient suffered from COVID-19 symptoms, including fevers and difficulty breathing. They each received a single infusion of mesenchymal stem cells sometime between January 23 to Feb 16. A few days later, the investigators say all the symptoms disappeared in all 7 patients, and they reported no side effects. So, stem cell proved helpful in a small group of patients suffering from the current pandemic, COVID-19. Monoclonal antibodies. As for monoclonal antibodies, an antibody produced by a single clone of cells or cell line and consisting of identical antibody molecules. These antibodies are known as monoclonal antibodies. Milstein and Hullo made first monoclonal antibody to target neurotransmitter in 1975, between 1975 to 79. The figure shows the hybridoma technology. Here, what do they do to produce monoclonal antibodies? They challenge mice um, with an antigen, and the spleen starts uh, making B cells specific, which produces specific antibodies. In the meantime, they took myeloma cells, which are cancer of B cells from cancer patient, and they fused both type of cells to make hybridomas, and culture in hat medium selects for positive cells. Positive cells means that the cells in hat medium, the cells which survived in hat medium, spleen cells from mouse died, and myeloma cells will die out because of some constituents present in the hat medium, and only cells which will survive will be hybrid cells, which will have uh, properties of spleen cells, that they will secrete one type of antibody, and myeloma cells, which are immortal cells. So only hybrid will survive in hat medium, and it will now produce only single kind of antibody, which is known as monoclonal antibody. So we use hybridoma technology, which the Milstein and Tullo used for the first time. Later on, it was patented also. The method was patented also. Uh, in August 75, Kohler and Milstein moved to patent their monoclonal antibodies. After this, research accelerated in this field, and major events that followed in recent years are as follows. First monoclonal antibody therapeutic for cancer approved for market in 1995. First fully human monoclonal antibody drug approved for market in 2002. We have seen that initially it was made from mouse. Now monoclonal antibody shown to effectively cut cholesterol level, thereby preventing heart attacks and strokes, was shown in 2017. And the first patient treated in first clinical trial using monoclonal antibody drug to treat schizophrenia in 2019. This was the advancement in the field of monoclonal antibodies. Monoclonal antibodies, which are actually used for therapy, uh, are lab produced molecules engineered to serve as substitute antibodies that can restore, enhance, or mimic the immune system's attack on disease cells of the host. They perform various functions, including tagging disease cells for the immune system to fight, triggering cell membrane destruction, blocking cell or blood vessel growth, and blocking immune system inhibitors. Since monoclonal antibody treatment has fewer side effects than traditional chemical therapy, treatments. It is better alternative to chemotherapy or as a parallel treatment for, especially for cancer patients. This is the INN system of recombinant monoclonal antibodies. This is completely human antibody. This is humanized. Second one is humanized antibody, in which other part, the antigen uh, capture part is mixed with some other uh, antigen. Then chimeric antibody, where human antibody, various parts are, main parts are from human being, and the antibody antigen capturing part is from mouse. And the last one is the murine monoclonal antibody. This is totally murine monoclonal antibody. The naming system of therapeutic antibodies, if it is some prefixes are used in in uh, in front of map, like U for human, O for mouse, A for rat, E for hamster, I for primate, XI for chimeric, ZOO for humanized, AXO uh, is for rat mouse hybrid, and this is FU for fungal. This is according to WHO guidelines. FDA approved monoclonal antibody drugs, and at least there are several, at least there are 20 drugs which are approved uh, for various trials, and each monoclonal antibody has a role in treating a targeted disease. For example, some drug treats transplant rejection, while other treats SLE disease, and all do something or treat some uh, condition. Treatment of COVID-19. Use of monoclonal antibodies. Reports say that in COVID-19, cytokine levels are increased. So elevated levels of IL-6 were found to be a stable indicator of poor outcome in patients with severe COVID-19 with pneumonia and ARDS. One clinical trial drug using the IL-6 receptor targeted monoclonal antibody, tocilizumab, reported quick control of fever and an improvement of respiratory function in 21 patients with severe COVID-19 treated in Anhui, China. All patients, including 2 who were critically ill, have recovered and have been discharged from hospital. So this was the progress in case of COVID, when monoclonal antibody succeeded in treating the patient. Now, genome sequencing. What is a genome? Genome is an organism's complete set of DNA, including all of its genes, that is from mitochondria, and if it is the case of plant, then from chloroplast also. Complete genome, complete set of genes present in an organism is known as genome. Genome sequencing is the serial sequence and composition of nucleotides present in the genome. What is whole genome sequencing? Whole genome sequencing is the process of determining the complete DNA sequence of an organism's genome at a single time. This entails sequencing of all of an organism's chromosomal DNA as well as DNA contained in mitochondria and for plants in chloroplast. Uh, this is the scheme of DNA sequencing. DNA is extracted, then it is fragmented, then it is cloned into vector. From bacteria, uh, these vectors are introduced in bacteria, and then from here uh, they are taken out after the growth of bacteria in large number, and this makes the sequence the library, make a sequence of all these fragments, and these fragments are then sequenced, and after sequencing they are um, assembled together. These are known as contiguous fragments, and these are assembled and aligned to give rise to a complete DNA. Pre-genome era. Pre-genome era means when we didn't know the genomes. Genome sequencing started with the Human Genome Project. This was launched in 1990, and researchers predicted that within 15 years uh, we will have the complete human genome, which consists of 3 billion letters of nucleotides. During this period, not only human genome was sequenced, uh, other organisms were also taken by different projects, and first bacterium genome, even before the human genome was sequenced, was complete, and that was of Haemophilus influenzae in 1995. Then Saccharomyces cerevisiae in 1996. Then C. elegans, which is a model organism. C. elegans, and then the model organism Drosophila was the genome of Drosophila was sequenced. Then Human Genome Project was completed in 2003, and it announced they announced that human beings contain between 20 to 25,000 genes. Genome and post-genome era. Now, with the completion of sequencing of human genome, we entered the era of genome and post-genome also, because after that they started to study genome. Initially, we had the sequence of genome only, and then they had to decide the structure and function of genomes, and various parts of genome must be analyzed. So we entered the genome and post-genome era. For some organisms, it was still pre-genome era, and for some it was post-genome era, because their genome was the information of their genome was complete, and their genes and their structures were studied. Other important genomes sequenced were of mouse, rat, chimpanzee, etc. These are important because these are mammals, and these are related to us also. That is why these are their genomes were studied first. Cancer genome was published, including lung cancer, malignant melanoma. Later on, in around 2011, three major new sequencing platforms were released, uh, which were PGM, Personal Genome Machine, RS and Illumina in 2011. Genome sequencing nowadays. What is the condition or a status now? Genome sequencing is a technology which has advanced to the point that thousands of genomes have been fully or partially sequenced. Doctors could not even now practice personalized medicines because most diseases occur due to problems in more than one gene, and uh, polygenomic defects create problem, while monogenic defects are easier to treat. Human genome research still continues to analyze different genes and their mutations, and the technology has entered the private market also. Companies like 23andMe or startups such as Helix, Veritas, and Color provide the public with lab-produced genome sequencing tests to see, discover ancestry information, or whether individuals are susceptible to heredity-linked diseases. Now, genome therapy, gene therapy. Once genome is sequenced and the structure, function, and mutations in genes are found out, gene therapy may be planned. Personalized medication is possible in theory. In medicine, gene therapy is defined as a therapeutic strategy that transfers DNA to a patient's cell to correct a defective gene or a gene product in order to treat diseases that are not curable with conventional drugs. 2017 marks as an important year of gene therapy and is considered as a launch point for a new era of modern gene therapy. Uh, One of the method of gene therapy is that modified DNA is injected into virus, virus which is acting as vector. Then vector binds to the cell, uh, getting a gene therapy. Virus leaves its protein port outside, and its genome or its whatever genetic material it has is injected inside the cell. It goes down and integrates into the host DNA. From here, it replicates and makes a large number of viral genome, and that is again coated by protein, which it produces itself after integrating to host genome, and then the complete virus uh, particles are formed inside the host, and they rupture the cell membrane and come out of the cell. Now, for therapy, we can use modified DNA. That modified DNA is injected into the vector. This is how modified DNA reaches the host DNA, where it is integrated, and then that is replicated, that is transcribed, and various proteins of therapeutic use are produced. This is the mechanism of action of this type of gene therapy. Gene therapy drugs. The future of medical biotechnology is certainly intertwined with gene therapy drugs. Nowadays, many sufferings from genetic diseases can be cured, especially when it is due to monogene defect. Multiple gene defects are complex. Viral vectors, mainly retrovirus, lentivirus, adeno-associated virus, in comparison to non-viral vectors, are the preferred gene therapy vehicles in the clinic. High efficiency of gene transduction, specific gene delivery and targeting, safety and reduced administration dose are the main benefits of viral vectors when they are used for gene therapy drugs. Approved human gene and cell-based gene therapy products. In vivo approved gene therapy drugs, which is a direct in vivo administration of manipulated viral vehicle for gene delivery, as I shown in the um, uh, figure. These drugs are a number of drugs approved are written here, and two of these are injected directly into the target tissue or organ. Another variety of drugs are ex vivo gene therapy, in which stem cells get genetically engineered DNA, and then stem cells grow into a variety of cells with that changed genome. Again, these are the names of various drugs of this variety. Latest gene therapy. In December 2017, the US Food and Drug Administration approved new gene therapy treatment for a rare genetically inherited form of blindness. The therapy called Luxturna is the first directly administered gene therapy approved in the US that targets a disease caused by mutations in a specific gene. The therapy works by delivering a normal copy of RPE65 gene directly into retinal cells. This helps the cells to produce the normal enzymes that help convert light into sight. So, actually, one operation has been done to, and patient got normal vision. So, this is the latest in gene therapy. Now, CRISPR technology. This is another latest technology in the field of biotechnology. What is CRISPR? CRISPR is clustered regularly interspaced short palindromic repeats, which are the hallmark of a bacterial defense system that forms the basis of CRISPR-Cas9 genome editing technology. This is basically a genome editing technology which was originally present in bacterial defense system. This is being used to help human beings as gene editing tool. CRISPR Therapeutics is founded with the mission of developing gene editing based therapeutics for serious diseases in 2013. CRISPR technology is a simple yet powerful tool for a CRISPR, pronounced CRISPR, strands editing genomes. It allows researchers to easily alter DNA sequences and modify gene function. Its many potential applications include correcting genetic defects, treating and preventing the spread of diseases, and improving crops. Mechanism of action of CRISPR. This is the DNA, target DNA sequence, which needs correction or which must be modified. Then CRISPR is used in the form of one guide RNA. This guide RNA can recognize the target sequence on the DNA, and here Cas9 is an enzyme which is CRISPR associated protein. This Cas also binds there, and it is capable of cleaving the both the strands of duplex DNA. As soon as it is cleaved, the body knows that DNA needs repair. Now, there are technologies to introduce desired genes at this point, at the cut point of the host DNA, and this is how mutation is introduced in the host DNA, and this mechanism is used as gene editing and gene therapy method. Examples of application of CRISPR: researchers in 2017 at University of California, Berkeley, developed a new version of Cas9 gene editing technology called CRISPR-Cas. This technology uses gold nanoparticles for delivering CRISPR-Cas9 gene editing system to cells that, when tested in MDX mouse model of Duchenne muscular dystrophy, repair the faulty DMD gene, leading to improved strength and agility. While CRISPR has already been used to edit human cells outside the body to treat cancer, blood disorder, and HIV, it was announced that the first ever CRISPR gene editing within the human body has been carried out as KCI Institute at Oregon Health and Science Institute, Portland, in March 2020. Diagnostic test for COVID-19 using CRISPR technology. There is a report by James P. Brugton et al. In this year also, this year only, they developed a rapid, easy to implement and accurate CRISPR-Cas12 based lateral flow assay for detection of COVID-19 from respiratory swab RNA extracts. This CRISPR based detector assay provides a visual and faster alternative to the US Centers for Disease Control and Prevention COVID-19 real-time RT-PCR assay with 95% positive predictive agreement and 100% negative predictive agreement. There's another report of use of this technology, and this report was given by Dr. Chakrabarti from ICGB, IB Institute in New Delhi, that Indian scientists have successfully developed a low-cost pay-per-strip test which can detect the new coronavirus within an hour and address India's urgent need for rapid testing. This test uses the cutting-edge gene editing tool CRISPR-Cas9 to target and identify the genomic sequence of the novel coronavirus in the samples of suspected individuals. This is a great news for us because we need diagnostic kit very urgently in large number too. So this was a very cheap modification of alternative of RT-PCR, and it takes less time also, so that in less time more population could be diagnosed or tested for COVID-19. As for therapeutics, a new Cas13 RNA screen has been used to establish guide RNAs for the COVID-19 coronavirus and human RNA segments, which could be used in vaccines, therapeutics, and diagnostics. And a novel CRISPR-based editing tool is also there that enables researchers to target mRNA and knockout genes without altering the genome. Using this CRISPR-Cas13 enzyme, Researchers have created a genetic screen for RNA, currently designed for use on humans, which they say could also be used on RNA containing viruses and bacteria. So, conclusion, in view of our advances, I can conclude that since each year scientists and medical biotechnological researchers advance further into the field of stem cell research, in future it might also be possible to regrow human cells, potentially replacing limbs, organs, or even healing cancer patients. Stem cell therapy can be used for COVID-19 patients also. Monoclonal antibodies can be used as therapy for diseases, and it has been used for monoclonal for COVID-19 also. In case of single gene defects, it is possible to do corrections if the structure of that gene and its defects are known. Diagnosis of COVID-19 using CRISPR technology might help us to screen population quickly and provide treatment to infected patients in time and get rid of devastating pandemic. They are searching the possibility of gene therapy and vaccine also using Cas13, which is again a CRISPR technology. These are the references I consulted, and thank you.